forward here. Huh? Welcome to the Ph.D. defense here today at the University of the Faroe Islands. Welcome to our Ph.D. student, Inga Kristiansen, who will defend her Ph.D. thesis with the title Population Dynamics of Calanus Species Within the Southwestern Norwegian Sea, Links to Water Mass Distribution and Norwegian Spring Spawning Herring. In the Faroese version, it's called Sambandet mellom djóraæti, havstreymar og norðursild. And very welcome to our panel of examiners. We have two of them with us here on campus today. It is Assistant Professor Annie Durus from the Faculty of Science and Technology here at the University of the Faroe Islands. She is the chair of the panel of examiners. And the first opponent, or first examiner as we used to say, is Hein Rune Skjoldal, who is scientist at the Institute of Marine Research in Bergen. And online we have Jeffrey Runge, who is professor of oceanography, School of Marine Sciences at the University of Maine. So I hope Jeffrey can hear us and see us here today. The agenda is, as you can see here, first Inga will have the opportunity to present her thesis. She will give 30 minutes to do that. And then comes first examiner, Hein Rune Skjoldal, who will do the first examination of Inga. And after that, we will have a coffee break. And during the coffee break, we have an opportunity for ex auditorio questions, meaning that you and the who are here can have questions to Inga. And if so, you need to relay the questions to me during the coffee break. So just let me know if you have any questions during the coffee break. And if there will be ex auditorio questions, then we'll take them just after the coffee break. And after that, and also if there will be no ex auditorio questions, we will give the floor to Jeffrey Runge, from who will be the second examiner. And after that, we will have deliberation, which will take about up to 20 minutes. And the chair of the panel will come and tell us the decision of the panel of examiners. And hopefully after that, the candidate will receive the diploma. So I'll give the floor to Inga Kristiansen and to have the opportunity to present the work. The floor is yours.
Let's see. Yes. Thank you for the introduction. Yes, so my name is Inga, and I'll present to you today my PhD thesis. So for the last couple of years, I have been focusing on these tiny little copepods here, which we call Calanus species. And I've been looking into the population dynamics of these within the southwestern Norwegian Sea. Some facts before we progress. Then there are two different types of these Calanus individuals. We have Calanus vermarchicus, and we also have Calanus Hyperboreas. So I will just refer to these as Calfin and Calhip. And these are only a few millimeters in size. However, they are found in huge amounts within the Norwegian Sea and also the Iceland Sea. And they are also key prey for herring, who I will also talk about. So after herring, when they finish spawning along the Norwegian coast, it migrates into the Norwegian Sea, searching for these tiny little individuals to prey on. One of the motivations why this work started was because there was a decline in zooplankton biomass around in the early 2000s. And around the same time also, there's increased numbers of herring that were feeding within the Norwegian Sea. And not only did their numbers increase, but they also fed for a prolonged period within the Faroese waters. So this caught attention, and this is one of the reasons why we started this work. So the results of the thesis is presented in 5 papers. There are 4 papers that I am the main author of, and I am the co-author of 1 paper. 4 of them have been published, and when the thesis was submitted, one of them was a manuscript. However, now this has recently been submitted to a scientific journal. And today I'll be talking about the 4 papers that I am the main co-author of, or the main author of. So first, I will talk a little bit about the seasonal cycle of calfin, and then I will progress into the interannual changes that we have seen of this individual. This will be followed by to talk about zooplankton and what influence water mass distribution have or how water mass distribution can affect these species. And then finally, I will talk about the feeding conditions for herring. So these copepods, both of them perform seasonal vertical migration. So during winter time, they reside at great depths. This can be up to 1,000 meters depth or so. However, towards spring, when favorable conditions are returning up in the upper layers, they migrate upwards, they molt into adults, and then they start reproduction. And this reproduction and stage development, that continues throughout the summertime. And then towards the end of the summer, they can decide whether if they want to use the energy that they have assimilated through food intake, if they want to continue a second generation, or they can decide to migrate downwards again. And here, they will remain in diapause until the following winter. Or, I'm sorry, the following year. So, as we see throughout their life cycle, they use the entire water column. So when they're at depth, these are transported with deep currents, and when they're in the upper surface, they're transported by the upper surface currents. So my main study area has been within this green box. But in order to understand what process is happening within this region, we also need to know a little bit what happens outside of it. So here, furthest north, we have polar waters flowing southwards. And a branch of this polar water enters the Iceland Sea, where it mixes and becomes the East Atlantic Current. And that current is transported towards the Faroes. And then from the south, we have warm Atlantic waters coming northwards. And these continue further into the Norwegian Sea. And then this inner branch here becomes recirculated back towards the Faroe Islands. So between these warm and cold water masses, we have frontal regions. And one of them that I'll be referring to quite a bit, this will be the Iceland-Faroe front. And the other frontal area here, this is the Yamain front, which is also important. In addition, we have the Norwegian sea gyre, which, as you can see, it circulates in a anti-clockwise circulation, similar to the currents. And this is highly influenced by the winds. So when the winds are strong, the acceleration of this Norwegian sea gyre increases and thereby is able to move frontal boundaries and is able to move this colder water more eastwards towards into the Norwegian Sea. So just bear that in mind for later. We also have our a study area that we call Section N, and this is a section that two of my papers are more or less solely focused on. And along this section, we have colonist individuals coming from the warm Atlantic waters. So these are coming from the south, and they are within the southern region of Section N. And then from the north, we both have colonists coming from the Iceland Sea, and we also have them coming from the Norwegian Sea and Iceland Sea. And in addition, we also have this Kongshøll, which is an Arctic species, and it originates from the Iceland Sea and is directed with the current towards our northern part of our section. And this section, it has been monitored since '93. It was established by Havn, the Faroe Marine Research Institute, and it was established in order to be able to monitor the ecosystem within the southwestern Norwegian Sea. And data that have been collected are data on zooplankton and on ocean characteristics such as temperature and salinity. Right, so if we first talk about the seasonal cycle of Kalfin, we did this seasonal study from July 2013 to July 2014. And so we compared conditions south of the front and north of the front. And you can see here that the red boxes, that's in the warm water, and the blue boxes there from the north. And we see that the female sizes, they are larger in the north than they are in the south. We also looked at the egg production of these within these two areas. And I am just focusing on April and May data. And here we can see that the spawning frequency. So the spawning frequency is the number of females that are producing. And we see that is quite similar between these two water masses. However, if we look at eggs per female, so how many eggs each female is producing in these individual or in these different areas, we see is quite different. We see that the females in the north, in the colder water, are producing much more rather than the females from the south. And this can be because of the increased female size. However, we do also know that food is important for egg production, so the more the females they have to eat, then they're also able to put more energy into their egg production. So we also looked at the food conditions. And here we saw that the females, they had actually quite high gut content compared to the northern females. So which was a little surprising. We also looked at the food availability for these individuals within the south and the north around the same time. And here we saw that in April and May in the south, there was not a whole lot of food for them to feed on compared to what the individuals had in the north. So these were some slightly conflicting results, and which we concluded that could be studied further. Yes. So this was, these studies were conducted at Section N, and the interannual studies, they're also from Section N. And this is data from May, since '93. So we see the years here. And again, we are comparing how the stage composition looks in the warmer water compared to the northern water. So this plot shows the stage composition in the Atlantic water, and I have grouped the developmental stages into 3 different groups. So the green, that represents the recruits, which are the youngest individuals, and then the orange, those are the juveniles, and purple are the adults. And there are some years where we don't have any data from, which is indicated by the crosses. So here we see that each year in May, there's a dominance of the recruits. So this means when we sample here in May, that the population has already started to reproduce. If we look at the north, in the colder water, we see that the dominance is different. So here we see that the juveniles and adults are dominating. So these are the individuals that have migrated upwards towards the surface, but they haven't started to reproduce yet. But then in 2003, we saw this change. And now, all of a sudden, this pattern here is very similar how it looks in the Atlantic water. This time series here is updated from when it was published. So we do actually see in the recent years, we see a change again back to the old pattern. But I'll get back to that. And for now, we just want to ask the question of what happened after this 2003 shift. So we looked into different possibilities. One was, we know that the temperature increased throughout the Nordic seas. So we looked into temperature to see if that could have increased the developmental rates. But this only shortened them by a few days, so this couldn't really be a possibility. And then we looked if the feeding conditions had changed somehow. And here we can see when the bloom onset starts. So here, and this is the day of year. And blue represents that here earlier, the bloom onset is earlier compared to the warmer water. And if you look through the time development, we see there's no particular pattern in the subarctic water that could relate to this change that we saw in stage composition. So in that paper, we started wondering whether if it could be a change perhaps in the water mass distribution. And that led to the following paper. And the reason why we started to think this is because we also have a time series of Kallhip. And remember, this is the individual who's an Arctic species from the Iceland Sea. So up until 2002, we had quite a bit of Kallhip present in the subarctic waters. However, in 2003, they more or less disappeared. And then if you look at the older Kallfin stages, so these are the juveniles and the adults, we see that their pattern is quite similar. So we started developing this working hypothesis whether the disappearance of Kallhip and large Kallfin, if that was related to the reduced influence of East Atlantic water. And East Atlantic water, that is affected with the East Atlantic current. So the idea is, so here you have the different water masses associated with the currents that I showed you earlier. And as this East Atlantic water approaches our section N, it becomes increasingly mixed between these two water masses. And this establishes a new water mass, which is called the modified East Atlantic water. And as I showed you before, we have Kalnes coming from the Norwegian Sea and Iceland Sea, and in addition, we have Kaldhøp also coming from the Iceland Sea. So the idea behind this hypothesis is if the East Atlantic water would retract, then it would also take its species along with it, and thereby resulting in a different stage composition here. So to test this hypothesis, we need to have a larger data set, and we used the zooplankton biomass over the Norwegian and Iceland Sea, which is collected during international ecosystem surveys in May. So here we have the mean biomass from 98 to 2016. So we actually don't know in this map what individuals are here, but we know that Kalfin is the most numerical dominant individual within this area, and in terms of biomass, Kaldhøp is dominating within the Iceland Sea. So these are well represented here. And we see that we have huge abundances within the western region and is diluted further eastwards. And we also see that in the warmer Atlantic water, there's much less concentration of zooplankton. But we saw this change in 2003, so we wanted to see how we want to calculate the difference before versus after 2003 in order to see where the largest change had occurred. And that we can see, so the blue represents where the largest change occurred, and we can see that this is just within the pattern of this East Atlantic water. So we wanted also to see, throughout this period, we wanted to see a time development. So we chose an upstream area respecting the current boundaries here, and we also chose this downstream area. And we saw that in 2003, there was a sudden change within this area here. And we also see this pattern of high abundances and low abundances. And when we look at the upstream data, there's a similar pattern. However, we see that the change occurs 1 year later. And this can be explained as a lagged effect of 1 year. So the changes would first occur here downstream, and then the following year, the East Atlantic water has retracted further northwards and thereby taken the individuals with it. So we are seeing now a change in the biomass, so is there actually a change in the ocean climate? Here we have the East Atlantic water. This is temperature at 100-meter depth. And this is in 2002. We have our section, and we can also see that this is the modified East Atlantic water that's extending past our section. And this is also a year when this Norwegian sea gyre had increased its circulation. And then if we take a contrasting year in 2003, we see there's much less blue, and there was also a year when the circulation of this gyre here relaxed. This is an image, again, of our section. So this is closest to the Faroes, protruding northwards into the Norwegian Sea. We have our different water masses here, and we have our frontal region. So we used the modified East Atlantic water as a proxy in order to determine if we could see any changes. And doing that, we took the area what this water mass occupied each year at our section, and we produce a time series. And in this time series here, you can see that there's a drop in 2003, and it's also characterized by these higher influx period and lower influx period, which is pretty much similar to our characteristics that we see in our Kelvin data. And what is quite interesting also is that since 2017 or so, we have seen an increase in this water mass again at our section. And this has also coincided with an increase in the zooplankton. So, which is kind of neat. So it just kind of proves that our theory, it holds that there is association between these two effects. So, because we have seen these changes, these bottom-up changes, and we know the herring, they prey on zooplankton, we wanted to see if the herring was influenced by this. Here we have a temperature map of the region, and Eliasen, she recently got a paper published, which I am also a co-author of. And here she showed that the center of gravity of the oldest herring has been very much localized within this region here since 2005. Earlier this year, Hatun, he also published a paper within this area. And here you can see the path of the East Atlantic current. And what you can see here is not just free flow towards our section N. Rather, the current, it goes very closely along the Yamine Ridge, and then follows it very closely southwards, and then it veers around this pointed nose here, and then it's free flow. So it kind of appears that there's this main source of inflow of these individuals would be just around this area. So because of these changes that we saw, we wanted to take some extra samples in May 2020, look at the zooplankton composition, and we also wanted to look into the diet intake of the herring. So in 2020, we saw that the zooplankton biomass was highest within this area where the herring is concentrating at. And the individuals that were responsible for this increased biomass here, that were the Kelvin, the larger individuals of Kelvin and Kallhøp stages. These two plots, they show the herring stomach fullness index, so how much the herring has been eating, and this is in different periods. Here we have the lower period from 2007 to 11, and then from the later period with an increased influx from 17 to 20. And we see two things here. First of all, we see that the further west the herring migrates to, they have increased stomach content. And second, we see that the herring has been eating much more here in the recent years, during the higher influx years, rather than in these years here. We also have data from the mid-90s, and Eliasen in this paper, she showed that from 96 to 98, herring was quite localized within the eastern side along the Yamine Ridge. And then from 1999 to 2004, it was localized much more northeastwards within the Norwegian Sea than previously. And then from 2005 and until present, we see this this southwestern position here. And then we can ask ourselves, why does the herring change its distribution pattern? And this is data from May, so this is during their feeding period, or the early start of their feeding period. So one thing that we looked at, well, you can look at many factors, and we chose to look at temperature and food availability. And as for the temperature, we produced a time series here within the East Atlantic Current. We got data from people from Iceland. Here we have temperature, and here we have the salinity. And what we can see here is that in the from 96 to 98, there were polar conditions observed. So during this period, it was very cold, and this is likely why the herring remained east of the Yamine front. However, since then, we have seen an increase in temperature. If you look at the zooplankton, then we see during these earlier years, during the higher influx years, there's plenty of zooplankton available for the fish to eat. So it seems to have a positive effect. If we look at when the zooplankton concentration started to decrease, the herring started to migrate southwards again within this position here, and has remained here. And we have divided these years up into two because of the recent findings that we have seen at our section, that we have seen this increased influx again. And we would have expected the herring to move further eastwards because of more appeared Arctic conditions. But when we looked a little bit further, we saw actually the temperature is actually quite suitable for the herring. So now they have this added benefit of remaining close to their feeding source here. So we have shown that section N, it contains two separate kelphin populations. We have shown that the 2003 reduction of cowhip and large kelphin, this is caused by a retraction of the East Atlantic water. And we have shown that the influx of the East Atlantic water has a positive influence on the herring diet intake. And before I conclude, I just want to acknowledge funding sources from the Danish government, from Félagið Nótaskip and Havn and Fiskvinningsgranskning, because without their funding, this work wouldn't have been able to be conducted. Also for the University of the Faroe Islands, for arranging this, for the committee and the opponents to taking their time to read my work and being here. And also for Havn, for colleagues, for all their help and support in many ways. And also, finally, my supervisors, Eilif, Chalmar, and Sigrun, because without your support, I wouldn't have been able to do this either. Thank you.
Okay. Well, good afternoon. Inga, you gave a very nice presentation, and I learned something new from your presentation today. So, dear audience, family, friends and colleagues of Inga, and representatives from the University of Faroe Islands. This is a big day for Inga. It's a milestone day, getting, hopefully, the Ph.D. for this thesis. Inga have now presented, and I will go back to some of the results. But broadly, it is a thesis with three main parts. The mid part is on the population dynamics of two of these Calanus species, Calanus finmarchicus and Calanus hyperboreas. And then on the other side, you have the ocean circulation and the spring phytoplankton bloom, or the spring phytoplankton, which is the food for these copepods. And then on the other side, you have herring, another fish, who feeds on the copepods. So this thesis is dealing with this chain from physics, copepods, fish. And as such, it is a broad theme, and a broad theme that Inga has treated in a very good way in these various papers that we heard her present now. The theme is broad, but yet in Inga's thesis, these are brought together in a balanced and good way. And so the committee is quite pleased with the thesis, and in my examination now, I will focus primarily on the Calanus and then the Calanus finmarchicus story, which are covered then in papers 3 and 1. And you showed some of these results, and I want to go back to some of the results from paper 3, so that maybe in a dialogue, in a conversation, we can go through that and see. And I'm particularly interested in sort of going through the generations. You have the overwintering generation, the G0, you have the spring generation, G1, and then you have the issue of a second generation. So I hope we can end up with sort of a clear agreement about the evidence for a second generation, which is a shift from previous literature. When I grew up and was younger, there was always sort of the idea that there were only one generation, and now a shift to two generations might represent a very major change in the ecosystem. I will also address then this issue of the spring phytoplankton bloom and have some consideration of how that the information you are presenting on the spring bloom, how that relates to the Calanus populations. And then in the end, maybe briefly on the ocean circulation and how it relates to Calanus finmarchicus in particular. Then Jeffrey, after the break, will then supplement what I have, or what we have been examining, but also going into the issue of the herring and papers 4 and 5, and possibly also touching upon Calanus hyperboreas. So this is result from your paper 3, and you have an upper panel here, Atlantic water with the absolute values and the relative values for the stage composition. And you have the various stages, copepodid stage 1, 2, 3, 4, 5, and then the adults, female and male. And you, Inga, you called the 1, 2, and 3 for recruits, and you called 4 and 5 for juveniles, and then you have the adults, the females and the males. So I thought we could go through this diagram, and it is a diagram from the 1,000 meter. This is the study in 2013, 2014, sampling with a multinet from 1,000 meter, and the multinet has separate nets, so you get a resolution of the vertical distribution in the water column. And then the dates are, this is July and August in 2013, and then it goes on with February, April, May, June, and July 2014. So we have 2 Julys. We have July 2014 and then July 2013 and the August. So I'm suggesting that we start with the February and the February situation in the Atlantic water. And just to explain, these are the absolute values and these are the relative percentages. So we can see that there are quite few numbers in February, but the stage composition is clearly shown here. So Inga, could you just briefly say what we see in...
Yes, of course. Oh, is it on? Yes, okay.
Yeah.
Yes, I'm sorry, yeah. So in the February data, the reason why it isn't, or it's quite low, well, one thing is that there can be individuals that are located below 1,000 meters in depth. So it's not, we're not sure if we got the entire overwintering population in this sample. But it is relatively low, and you can see what it mostly comprises is the stage 5, which is the typical stage where who overwinters. And we see some of the we see also some females and males are present, so which means that they have molted into the adults stages here.
So the issue of you see the males here, and they are almost the same numbers as the females, and that is typical for early phase of reproduction, where we often see the males are sort of skewed to the early phase of the seasonal development. Mm-hmm. If we then go to the April in the same, what could you say here?
As for the April data, we see, well, now there are more females that are present. And these are the females who are responsible for the egg production. It appears also, we also have some of the younger stages that are present. So stage 1, 2, and 3. And the males are still there. However, I mean, the abundances are still not as high.
But a comment to these abundances, this is about 10,000, these values are 1,000 times number per meter square. So we have roughly 10,000 individuals, and that is a fair overwintering generation. You can see higher values, but it is not exceptionally low either.
No.
To be a...
Yeah.
So you would call this the C5 here and the adults, and the fewer C5, more adults here. This is the G0 population getting ready to spawn or spawning at this time. Is that?
And you can also see that from the February data, the numbers of the stage 5 that has decreased in April, so which indicates that they have molted into the adult stages there.
So if we then go to the subarctic water and take very quickly the February and April data here, there are somewhat higher values, maybe 10,000, a little bit more than 10,000 per meter square.
Mm. And yes, and we also see there's much more of the stage 5 that are presented here. and also there's less males and females, so it seems that the system in the north is a little bit slower, which is not surprising because of the colder temperatures.
But when you come to April, if you compare April here and April there, what would you say then?
Well, the number of females, they look is quite they're quite similar. And it appears also that here from the April data, we also have that egg production has started here because of the increased numbers of the younger stages here.
Mm-hmm. And then if we go to May, you see we have much higher values, and you get the recruits. You see the recruits here.
Yes, so it takes some time from so you see a much higher abundance of the females in April, and it takes a few weeks in order for these individuals to get to stage 1. So you can see now that this is the first generation that is properly appearing in this season. So the peak of reproduction in Atlantic water occurs in May.
Yeah. And then you see 1, 2, 3, 4, 5, and the adults. So
Yes.
how do you evaluate the adults here? Are they G0 or are they G1?
I guess the individuals, they are migrating up towards the surface at different times. So whether if they are individuals that have migrated up later in the season, that could be a possibility.
Yes.
Or potentially, I guess there are tiny amounts of the youngest individuals that are present in February.
But from the stage composition here, you have low numbers of 4 and lower numbers of 5, and then more of the adults again. My interpretation would be that these are G0. Like you started by,
yeah.
These are late arrivals. And,
The later arrivals.
And still.
Yeah.
So we don't know the the answer here, but it is an interpretation. So.
Yeah. And this, I think that was also my conclusion in the paper. That yeah.
I should say that in your thesis you have written about that and these things, and I don't think you have written anything wrong there.
Okay.
But I would like just to go through this so that we could see sort of a clear overall picture.
Mm-hmm.
So then we go to, we have a similar situation here in the subarctic water. Again, quite similar between the Atlantic water and the subarctic water.
Yes, it's very similar, yeah.
And then if we go to June, and then June in the subarctic water, something happened in this figure here because the yellow and the red is missing there.
Oh, yeah.
So you have cut the axis, but I think that the total should be 140,000, but you have the relative stage composition shown here.
Yes, yeah.
So what would you say then about June?
Well, I guess here from this image, I mean, you can see that in subarctic water, that the reproduction, it is, you can see the evidence of the younger individuals in May, but I'm guessing because of the lower temperatures, you don't see the peak reproduction or the peak in of the G1 until in July. So they're peaking earlier in the Atlantic water compared to the northern waters.
So these this would be G1.
So those are yes.
Yeah. And if we then go to July, you have the situation resembles a little bit the June situation. So how do you evaluate then this July situation?
I think when you come further into the season, it's a little bit difficult of what's the G1 and what's the G2. But we could also see, I guess if you judge it by the numbers, the females, they weren't producing as much, so therefore you would also get lower concentrations. And based on in June, there aren't as many females either available in order to produce equally high amounts that you see as you see in June.
So I think at least one possibility is that this is still mainly a G1, that you have a protracted sort of spawning period and a protracted time curve for the G1. But there could also be some G2 starting to appear here.
Possibly, but I would mainly, I think in July, I would assume that mostly would be from the G1.
Yeah.
And the appearance of a second generation is perhaps observed a little bit later.
Yeah,
Yeah.
I agree. So if you go to this July, it shows maybe a clearer picture that there are some C4, there are more C5s, and there are still some adults here in this situation.
Yeah.
So this could be then the G1.
This is the G1 now that have matured or have developed into the juveniles.
Yeah.
And now I guess they are in the position of whether if they want to go to start to overwinter.
Yeah.
Or if they want to continue to molt into adults and to produce a second generation. Yeah.
Yeah. And then if we go to August, then you start seeing younger stages here and you interpret them as
And those are the ones from a G2 yeah.
So in this, so the clear evidence for a G2 is in August, where you see these increased numbers at quite high densities of the young stages.
Yes, and I think it's probably also coupled with the picture that we are seeing in July, because we're seeing this increased amount of these juvenile of stage 4 and 5, which have the potential to produce a second generation.
Yeah. So, and then you have also in July, and to some extent in June, you see adults, including males, here. And this would be likely to be the G1 that has some individuals have matured and are then producing the G2.
And they're producing the G2, yes, yeah.
So I summarize this, and I think what we have gone through now, that you have a G0 from the overwintering, the dominance of C5 in February through the adults, and still adults that could possibly be G0 in May. And then you have the G1 from the younger stages in April, moving through to overwintering stages in July. And then you have the G2 maybe appearing to some extent in July, but appearing in higher numbers as younger stages in... Is that a fair summary of sort of...
A fair summary, yes, and nicely illustrated also with the arrows, yes.
Yeah.
Yeah.
So then, going to the vertical aspect. So these are the same data here, and I included the... now I can't see. These are the July 2013 these are the August 2013. And so what we talked about was the yellow is the C5s, and then in July we have some red adults, and we can see a vertical separation here in that many of the green and the yellow, the C4 and C5, have migrated down to overwintering depths, while you have these adults probably reproducing from G1 in the upper layer, and then producing this quite high abundance of the G2 when you come to August here.
Mm-hmm. Yes, that's correct, yeah.
So is it then fair to say that we see a second generation, but it is spawned by only a smaller fraction of the G1, so that most G1 individuals move down to overwinter as G1, and then a smaller fraction matures and produce a second generation in the upper layer?
In the upper layer, yes, that's correct, yeah.
And then on this vertical, you have the Atlantic water stations, and the Atlantic water is on top, and below about 300 meters you get down into the deep water. Mm-hmm. And I understand there is some complexity about these intermediate waters where they come from. But how would you characterize then the connectivity between these upper layers and the deeper layers for these two situations? I'm thinking about the ascent and the descent from these upper layers.
Okay. Well, I think I think the deeper water that is in the Norwegian Sea, I think it will be different from what's in the Atlantic water that is coming towards the southern portion of Section N. Are you thinking in terms now of direction where individuals are coming from, or how they are positioned, perhaps?
I was partly thinking that these generations, the G1 that develops up here in June and starting to migrate down, they would be then coming down into similar intermediate waters, maybe some differences, but at least they would go down into these waters in the same way as the individuals from the subarctic waters. They
would, yes. And so potentially that these individuals here, they would overwinter in the Norwegian Sea.
Yeah.
That's one possibility. But we also know that some of the deep water can also be expelled through the Faroe-Shetland Channel. Mm-hmm. So that can also be, and if they are expelled through the Faroe-Shetland Channel, then they're out of the system, at least for now.
Yeah.
Yeah.
If we then go to paper 1, and you explained this in your presentation, you explained this change. This is subarctic water. So between 2002 and 2003, you see this phenological shift. Mm-hmm. And you explained this is a G0. This is sampling from May. And then the green, the juveniles or the recruits, they are from the G1.
Yes.
Isn't that correct?
That's correct, yes.
And then the relative. Then you go on to September, and I'm citing here that you write paper 1 demonstrated that the phenology shift initiated in 2003 resulted in an increased numbers of young stages in late August, early September. When I look at these results, these are the September data in the subarctic water. This is 2003, and so there is no clear change just after 2003. So are you referring then to what happened subsequently, that you start seeing the green appearing from 2007 onwards?
Yes. So yeah. So we don't see the immediate effect, and the reason why we don't see an immediate effect, I am not sure.
No.
But it was somehow after this shift.
So what you have seen is a clear phenology shift in earlier appearance of G1 in the subarctic water in May.
In May.
And then you start seeing appearance of what is likely the G2 as younger stages in late August and September.
September.
Yeah.
Yes. Yeah.
Then you have a statement here, judging by the increase in abundance in stage C1 in August the previous year, suggests that a portion of the C5 molts into adults and produce a G2 in both regions. And this is what we have gone through. This contrast observations made by Strand et al. 2020 and references herein, who were only able to see a G2 within the Atlantic water in the Norwegian Sea. In your thesis, you refer often to Strand et al. As saying there is two generations in the Norwegian Sea, but here you seem to qualify that. So can you explain a little bit, or?
Yes. The conscious observation. Yes. I guess Strand, they are a little bit positioned a little bit further north. They're not just as south as where we are at our station. So the region which they have covered very nicely, they haven't, I guess, covered our region. Oh. But I agree perhaps that this, in this statement here, because based on the shift, and if we are seeing increased influence of this North Norwegian Atlantic water, I guess you can say rather that this within our region now we are also seeing a second generation here, which I guess is actually more comparable to what Strand is observing, and this is. But we have to bear in mind also that this is a warm period now that we're observing. So I guess we are just situated in this environment where we can be influenced by a G1 or a G2, depending on which state the marine climate is in.
Yeah, no, that's... And then you showed this, and you written some text here. The notable difference observed in February could potentially be due to the one advection pattern in relation to the position of the individuals in the water column, or two females who entered diapause from the year before. I found this a little cryptic.
Let me just read it.
Yeah. So I wanted to. Could you explain a little bit what you are thinking here, or is it?
Let's see.
You have the February that the females are much smaller
here,
and then subsequently in April they are comparable. So I assume you are sort of pointing to possible mechanism why the individuals were so small in April.
Well, we're not really sure why they should be as small in February if they're overwintering at great depths. And why there's such a large difference between, or just in February, whereas in April they become bigger.
I guess this the issue of also where does the females in the Atlantic water comes from. Some of them is probably coming from south of the Iceland-Faroe Ridge, being transported onto the ridge and then onto section N. And they are part of that complex with the outflux of Calanus, with the overflow water, and then they rise again and can become transported in the inflowing water.
Yeah, there are two different, or there are probably several different paths which they can come to our section N, Yeah, which is the southern portion. And that path it was difficult. I was trying to write about it, and I guess the most simplest way, of the most direct way in which they are directed towards our section, is probably just directly from the Iceland Basin. But the other way around, they can also circulate around the Faroes. Yeah.
And this is the lipids data. In the material and methods in paper 3, you write that the samples for lipids came from the same net holes as for the gut fluorescence, and that was the upper 50-meter holes. But clearly here you have data on the lipids from the deeper holes. So I assume these come from the multi-net samples.
From the multi-net, yes, yeah.
So they lack some information in the material and methods on that, I think. But I just noted this. We saw on this other diagram that in July and in August there was quite high numbers that had already descended from the G1 going down as mainly C5s. And yet they have very, very low lipid content. I put a question mark on that. I thought it was a strange result.
Yeah, and I think that's also one of the reasons why I didn't quite put that much emphasis on it when I was writing the thesis. Because if you compare also this data to other studies, the lipid content is quite low, and I'm not really sure why that is. I mean, it's quite unique that we have a time series which shows the seasonal lipid content, and I think it would be really interesting to do more of this research, so you can actually see the seasonal pattern of this. And I also think that this data here could be added with more data to see, if this is the true image of it. Yeah.
And then I think we move to this issue of the primary production or the spring bloom. And I think you showed also these two figures, or at least one of them, in your presentation. So you have the timing of the onset of the spring blooms in the subarctic waters, and here it is around day 120, so late April, early May, although there are some changes towards the end of this series. And then in the Atlantic water, it is later, around day 150, or late May, early June. So this is a clear difference based on the satellite observations and the onset. And then the data from the sea, you have from the May series, which is collected in mid to late May. You have these values then often lower in the Atlantic water, but still 0.5 microgram per liter. There is some error here in the units. But also up to 1.5. And these are quite high chlorophyll values. So when you state here that there is also a significant egg production during the pre-bloom period, which is before this bloom onset, you say, which is thought to be mainly fueled by lipid reserves. I don't think that need to be correct, because even if you say this is before the onset of the spring bloom, chlorophyll levels are still quite high. And if you compare to other studies, they are well within the amount where they can feed and produce, have a high egg production rates. So, and I think maybe this is related to the criterion for how you sort of, what you mean with an increase in or the onset of the spring bloom. So maybe you could say something about this criterion and how this is done.
Well, this was done by one of the co-authors, Sophia. And she had been developing this method for her Ph.D. thesis. And I think what she used here, so she used each seasonal cycle of the data, satellite data that was available. And then over the annual median, when there was a 5% increase in that, then that's how she got her value from.
But I would
But yes,
suggest maybe that this criterion is not that applicable for these waters in the Norwegian Sea, where you have a very large dramatic shift from very low values in winter to then higher values. So this is something at least you can keep in mind for future work to how to sort of address this initiation of the bloom.
No, I was just going to say one of the advantages of when you have this satellite observations is that you have only obviously in the surface, but you have much more data available compared to a snapshot that you have from the other time series that you showed from the right. So I guess it's good to complement both.
I agree fully with that. So, but it is a matter of then which criterion are you using for seeing that increase. This is from a box I produced in the Norwegian Sea Ecosystem book, and it's based on two studies by Halldal 1953 and Østvedt 1955 at Weather Station M. And these are Østvedt's data, and they are arranged on the same time scale as the upper one. So starting with the upper one, these are then the diatoms and the total phytoplankton. And we see this is a spring bloom that takes place in June in this data set. But prior to that, there is a pre-bloom phase in April and May where the concentration of phytoplankton increases to a modest level. But yet the Calanus development, these are the adults, these are the C1, C2, C3, etc. And this is the copepodite peak. And you can see that this peak occurs in this pre-bloom phase. We have also shown similar things in the Barents Sea and the Atlantic water. And I think this is an important sort of feature of the Norwegian Sea ecosystem, that there are a buildup of chlorophyll in the pre-bloom situation, which is sufficient for Calanus to spawn and to feed upon. As you show in your thesis, in April and May, there are egg production and spawning, and you see the resulting G1 from that spawning. And in the Norwegian Sea, this is a very effective system because it allows the Calanus population to develop in the pre-bloom period and during the bloom period and convert the primary production into secondary production in that. Then I will end by some consideration of the circulation. I noticed on your presentation earlier today that you had added this red arrow, because I added this. I think you should show that there is also a major inflow coming up through the Faroe-Shetland Channel. On other diagrams, you haven't shown it. You mention it in the text, so I know that you know about it. But for the completeness, it would be good just to show it. And you showed it in your presentation today. Then I added this question mark here. I understand that this ferrocurrent arrow is sort of a schematic, but could you say something? What happens further west here?
I apologize. You mean with the water? You mean Atlantic water transport?
Yeah.
Well, there are several branches that come from the south. I think the ferrocurrent, I guess that was just the main flow, but you have also one path here that is quite narrow along the Icelandic slope, and then it also curves to the
So coming up here and then...
yes,
yeah.
yeah. And then
Okay.
straight into a section, yeah.
So this might be a sort of a broader region. It flows over this, over a wider, and then it converges down here into the section.
I'm not 100% sure, but I mean, I think there are several branches of the water, of the Atlantic water that's coming
Yeah.
up, and where it's some parts where it's more focused, and then when they all come, I guess, together and north of the front, that is then when it becomes the Føroya current.
Yeah. And then you described also in your presentation today this, the recirculating currents of the Atlantic water, and you described this shift from the modified East Icelandic water and the not North Norwegian water, but Norwegian North Atlantic
yeah,
water.
Yeah.
You have mixed this sometimes.
Norwegian
in your,
North.
Yeah.
Yeah.
So I had some difficulty with it. You showed also this. You have the front, you have the Atlantic water, and then you have these two, the modified East Atlantic water and the Norwegian North Atlantic water. And then you write here, off the northeastern Icelandic shelf edge, the East Icelandic water slides isopycnically underneath the northward flowing modified North Atlantic water. Further east, the southward flowing NNAW submerges under the East Icelandic water and is wedged between the East Icelandic water and modified North Atlantic water. When it submerges under and then it is wedged between, isn't this a contradiction?
Let me see.
It's that it submerges under the East Icelandic water,
yeah.
and yet it is wedged between the East Icelandic water and the upper layer.
I guess as the as the modified East Atlantic water, when it becomes established, it kind of the water masses find their position, I guess, according to their densities. And this modified East Atlantic water, I guess, it kind of slides gradually further downwards.
So it can be a dynamic process.
So
in
it's
a sense.
so it's a dynamic process, and in this instance, you can see that the modified East Atlantic water is kind of in between the Norwegian North Atlantic water and the modified North Atlantic water.
Hjalmar can talk about this later
And
today.
he can probably talk about it for hours.
Yeah. Okay. I think this might be my, well, at least the second last slide. This is a summary where you show sort of schematically, you have the Calanus finmarchicus and the Calanus Hyperboreas coming from the Iceland Sea in this modified East Icelandic water. And then you have Calanus finmarchicus coming from the Norwegian Sea in this Norwegian North Atlantic water. And then you illustrate that you have a shift, you have a suppression, this withdraws to the west and is taken over here. You seem to imply that with this colder water here, you get one generation, but when you get this situation, you get two generations in this. We already discussed this, and also your reference to Strand et al. 2020, where you generally, when you refer to, they say that they saw two generations, but then you had that in contrast, which we qualified. But is it your sort of view that you, in general, have now two generations in this, or is this more a situation here in the southern part where, and I guess we don't know that, it's maybe that is something for your further work to look into.
Well, we have, we updated, we counted the data from May, but we haven't looked at the August data yet. So I guess it would be quite interesting to see if this recent shift that we have seen more of the increased Arctic water in the recent years, if we actually have seen the disappearance, or see whether if the G2 is still present or if it has disappeared. But that is something yet that we have data that we can work on.
Because going back to what we have discussed, the evidence for a G2 lies in that August situation and partly that September situation, where you see the juveniles or the recruits, the stage 1, 2, and 3, but it has not been documented yet whether the G2 has a successful recruitment or not to the overwintering population. So that remains to be looked into with future. Yeah. Then I think I have my last slide, and I didn't want to spend much time on this. You went through this in your presentation now. I just would like to comment on these values, which are extremely high biomass values, higher than 15, 20, up to 30 gram dry weight per meter square. Those are exceptionally high values. You have in paper 3 converting your counts of Calanus finmarchicus into biomass data. Do you recall what the scale on that, what the maximum on your scale is on that graph?
No, not right now.
It is 5 gram dry weight per meter square.
Yeah.
And typically, if you take counts of Calanus finmarchicus and convert it into biomass, you get values in that range, up to 5, rarely more than 5. So there has been much emphasis on this strong decline, and what you have been sort of suggesting, it related to the transport for the Iceland Sea, is a plausible sort of explanation for that change. But we should look more into these very high values, that what were really driving these. Calanus hyperboreas is probably one of the drivers for those high values, but there could also be other things in that plankton driving these very high biomass values.
That is true, and I'm guessing also when you have these more Arctic copepods or amphipods, I mean, they weigh a lot, and the more Arctic conditions there are, I guess you can also assume that these would be present in the sample.
So I have been looking at many zooplankton biomass data, and the only place I see similar high values are in the Bering Sea, in the slope waters, which have been called the green belt around the Bering Sea. So these values are exceptionally high. In the Barents Sea, we are sometimes seeing values in the northern part up into this range, 15 to 20. But and I think that ends my examination. And I think I kept within the time. So thank you. And then I guess there is a break, or?
Yeah.
Thank you very much. Now we have our break, a coffee break, and we will gather here again 14:40. And I should remind people about the possibility to put questions ex auditorio. So if there are any questions, so please let me know during the break. Thank you.
Hello, Jeffrey. Can you hear me? This is the technician. I'm just wondering if we could check the sound before we start again.
Hello, welcome back from the coffee break. There have been no questions from ex-auditorio. So we will continue right away with the second examiner, who is Professor Jeffrey Runch from School of Marine Sciences from the University of Maine. So Jeffrey, can you hear us and see us? We need to get this fixed. Hello, Jeff.
Yes. Yes.
You are.
Hello. I was on mute. Sorry.
Okay.
But I can hear you and see you.
Very good. So the floor is yours.
Okay. And do you see me on the big screen? Is that how it works?
Oh, I see you on the smaller screen there.
Oh, good. Okay.
Yep.
Yes.
Good. So first, I'm going to. Would like to share my screen in an instant, and I think I can do that. But first, I'd like to Inga, extend my congratulations for the work that you've been doing, and also to your committee for diving into the complexity of a very dynamic system there with and trying to make sense of the interactions between the physical circulation, the environmental conditions, the population dynamics of the foundation species in your system, and then relating that to the higher trophic levels, forage fish. Really enjoyed reading your work, and extend my congratulations for all the efforts that you and your committee have done. And also, I'd like to say that I'm sorry that I can't be there in person. I would really like to have been in Havn for this. So I will, while Heinruna was talking, I realized that I probably should expand my, it would be helpful in questioning that I expanded into a PowerPoint and take some of your slides and some of mine, and that would probably be more efficient as I try to do this virtually. So I'm going to share my screen now, and hopefully you can see this.
Yes, I can.
Good. And whoops, so let me just move back. So I'd like to start, I find this is a really interesting opportunity for all of us to explore and maybe get insight into the system that we share in the North Atlantic, the Calanus species being and their lipid supplies being such an important part in both systems. And so I'd like to start with just continuing a little bit of Heinruna's exploration of the reproductive response of Calanus, and just review some of the work we've done on the northwestern Atlantic for the reproductive response, the functional response, egg production that we find in the Gulf of Maine, Georges Bank. This is work that was done 20 years ago now, approximately, and especially, this hyperbolic response, and noting the egg production rates on the western side tending to be around maximum egg production rates somewhere around 40 to 60 eggs per female per day, and clutch sizes that range, depending on the size of the animal, from about 30 to 60 to 80 eggs per female in a clutch. And then, because it's interesting, there seems to be a contrast or differences between the Northwest Atlantic Calanus populations and the Northeast Atlantic Calanus populations in these reproductive responses. And here I've taken from the functional responses for egg production from Mella et al. For different regions. On the left-hand side are the Northwest Atlantic, Georges Bank, Labrador Shelf, Scotian Shelf, Rimouski, Gulf of St. Lawrence Station. In the Gulf of St. Lawrence, the filled circles are during the spring, early summer, and the open circles are reflective of the later other times of the year in places where Calendus is feeding not on chlorophyll necessarily, but on the microzooplankton in the low chlorophyll situation. And then on the eastern side, the right-hand panels, you have Iceland and the North Sea here. You know, the Icelandic pattern, the egg production rates seem to be kind of some similarity with the Western, but in other areas, there in northern and in WS, what's that? Northern Norwegian Sea, for example, the rates seem to plateau at a lower, at 20, I mean, half or so, although you do see some high rates. And I'd like to explore that a little bit with the results that you have. It's you know, trying to understand this the results better, and it also links with the slide that Hein Rune showed of a Norwegian Sea the ability of Calendus to reproduce prior to the bloom. So let's see what hold on a second. So looking at the results again, so maybe Inga, you could kind of review where you are in thinking about these results of between in the, you know, they sort of do fit in with the Northeast Atlantic results, where you have lower egg production rates and lower clutch sizes. You know, look at spawning frequency is close to 80% in April and May, yet eggs per female is you know, somewhere between 5 and less than 20. So that implies a fairly low clutch size per day and especially for what could be what's your thinking about why we have this, why we don't have more egg production?
Well, I know that the results that we see in the Atlantic water, I mean, they do fit with similar or with other studies that have been conducted within this within the same area. But I know that other studies, they have also done or explored more than I have as to why these egg production are so low. And I'm not really sure if they have found a reason why. I think there was one along the Iceland Føroya ridge. And they also looked at food composition, because food composition, that can, for example, you would have guessed that food composition that could perhaps influence how or the egg production. So if there are less diatoms, perhaps, then perhaps they're not as efficient to conserve or to change their energy into the reproduction pattern. But also, these individuals here, they're also smaller, and smaller females are also known to produce less. But if they have, but here in these results, here it shows that they have ingested quite a lot of food. So why that extra food content is not able to be put forward into the egg production rates, I'm not really sure of.
Yep, Yeah, exactly. That's interesting, so we can maybe hold that hypothesis of that there's food limitation, which I think is a really important. Thinking about factors that influence the dynamics of calonis in the system is this possibility of food limitation, and maybe just not necessarily quantity either, but the quality of the food, and maybe limiting egg production rates. I think that's something. And so I'm looking at I pulled your two figures here that you have, and just looking at them for April, especially the right-hand side here, the fluorescence, and looking at, I guess this is the Atlantic water and the East Icelandic water, so southern water. And in April, it looks like right around, so recalling that the critical concentration, or the concentration which you can get like even half max egg laying rates, chlorophyll concentration, spring, early summer, is somewhere between 0.5 and say 1.5 micrograms chlorophyll. Now this is fluorescence, and I'm not sure if that's exactly corresponds to micrograms chlorophyll A, but it does look like right at the right around 50 meters in April, and even in May, you're starting to see and I guess the bloom, the timing of the bloom metric that you developed, and I guess that the metric was 5% above median. Do you remember what the median value is for the 5% about what's the median value?
No, I can't remember that. I'm afraid.
Is it 1 or is less than 1? Yeah, any for that. I think that's important, kind of to understand the chlorophyll that where you can get substantial egg laying rate, and if you have enough females around, you can get substantial input of eggs into the water.
Yeah, we have this. We supplemented so the image that you have here to your right of the different water masses. So again, these are just snapshots that we have collected when we're out on our annual cruise or on our seasonal cruise.
Right.
And so the graph from the on the left, that the idea was behind that graph, that was to supplement the time development, when we're not out there sampling.
Right, and that's just from the satellite.
So this is just
the surface layer.
So it's just the surface layer, and also I think the satellites are also slightly you have to be aware of that they slightly underestimate also the concentrations. So these concentrations, they might be slightly higher, but being aware of that, we do see the general seasonal pattern within these two regions.
Yeah. So even so the satellite, I do wonder about, just surface layer that we're not capturing food availability deeper in the water column like we're seeing on these fluorescence profiles, even though it's not a lot. That it could be, and we see from the gut contents of calonis, or the gut fluorescence, that they are feeding. And so wondering if there isn't food there, it's just a question of, is it food limitation then why the egg laying rates are so low, or is it size of the animals? Or is it something, the contrast between the Northwest Atlantic and the Northeast Atlantic is maybe there's some genetic difference there too. Maybe there's some difference in the populations.
I mean, one idea with that, with the lipid data also was to be able to supply information on the lipids that the individuals had within these two different water masses. Because often, where individuals in colder waters, they tend to gather more lipids, are able to reserve it also better rather than individuals from the warmer waters.
Yeah.
So that time series, that showed us something, but I think it could have shown us more, or I wish it had shown us more. But that was also, I mean, that can also be a factor, obviously, influencing the difference in egg production.
Yes, and you know, it's interesting. Cold from the Gulf of Maine, which is at the very southern edge, 8 degrees to me is still cold. 4 degrees is Gulf of St. Lawrence. 8 degrees in the Gulf of Maine is relatively cold, and still seems to be in the optimum range for egg laying. But you know, so I was just wondering if, you know, so whether it's, you know, food limitation, whether there's possibly some, and I don't have an answer. I'm just kind of just interested to, you know, discuss or bring it up, that because I think they're all important, and I think the measurements of wax esters that you made in lipid are really important, and I would like to kind of come back to the role of lipids in the system later. But so there's food limitation possibilities. There's possibilities also that there are genetic differences between the eastern and western Calanus that I don't think have been explored. I also wonder about methodological possible sources of error in methodology, and especially the role of cannibalism that may be in the way that the egg production measurements were done. There are differences between the East and the Western researches often in how they were done. And
I'm sorry.
Wondering if my comment.
No. Just to comment on that. Then the method that we used. So we used this 1/2 liter bottle that we collected the females in, but we had this fake bottom where the bottom it was we had a mesh on.
Right.
So the idea is that you put the females into the bottle, and then the females would be on top, and then when they produce the eggs, then they would float down through the mesh. So I guess this is a way to minimize cannibalism, but we haven't actually tested the differences when you do use a mesh and when you do not use a mesh. But that was the method that we used, because I know there are different methods that they have done or they have used in Iceland and Norway and within your region also.
Yeah, I know that's a standard method, and you know, and we've used that ourselves. Erica Head did kind of a study of the methodology, and is it a 24-hour incubation for the egg laying that you do?
We did a 24-hour, yes.
So one thing I'm just throwing it out as an alternative to kind of look at and maybe, you know, keep in mind is, so when Erica Head did her methodology study, she did find that 24 hours, that even though the eggs are, you know, falling through the mesh, but there is a time when the eggs are suspended depending on where the females release the eggs into the water. And, you know, if it is a possibility that there is some cannibalism going on before the eggs even make it through the mesh. And there's, you know, I've done some of these experiments, and I actually found that using containers, I actually found that the sea state made a difference. And you know, we've been out in some conditions where the ship is really rolling around, and we find that I don't know if, you know, we have these containers. We did this way, containers, you know, with females suspended. I don't know whether it's you know, that this rolling, the sea state, actually helps keep the eggs in suspension, since they're only slightly negative buoyant, but we did find that there was a relationship between sea state and cannibalism in our containers that was quite concerning. Is one reason why we kept using dishes, little flat petri dishes, which is much smaller volume, individual females, and then they lay their eggs, and then the eggs rest right on the bottom, and it seems to make it more difficult for the females to get around them and eat them. But we still also went to every 8 to 12 hours, kind of measuring, paying attention, because you do notice cannibalism happening, even, as best we try. There's just the possibility of that as a source of error as well.
Yeah. We haven't looked into that, but that is something, yes, that we could be aware of.
Yeah. Okay. So, but interesting results here, and still doesn't explain the difference between AW and
no.
Yeah. So I wonder about you wonder about food limitation. I wonder about also that there are differences in the phenotypic, maybe genotypic responses of Calanus between the two sides.
I don't know. I mean, as far as I'm aware of, it's quite difficult to separate genetic differences within the Norwegian Sea. It's been a while since I've been reading about this, but I know they're genetically different from the Gulf of Maine, but these are quite separate systems. But within the Norwegian Sea, I thought it was a little bit more trickier to distinguish differences because of the evacuation pattern, and there's a little bit of a mixture. So being able to distinguish whether you come from the Iceland Sea or Faroe Shetland Channel is a little bit tricky. So I don't know if we would be able to do this within our section.
No, I agree. I wouldn't think that you'd find genetic differences between the section N and the Norwegian Sea. There's too much advection. That I would think they'd be homogenized. I was thinking more of the differences between the Northeast Atlantic and the Northwest Atlantic.
Right. Okay.
In general.
Yeah.
Yeah. All right. So go now on to Calanus Hyperboreas a little bit and understanding. And Hein Rune addressed this a little bit, and he showed this figure. And again, pointing out, I also am impressed by the high biomass in 2000, and assuming that's mostly Hyperboreas, and just wondering, do you have the, and this is in the upper 200 meters, is that correct, or is this 1,000? There's one that you had 1,000 to 1,000 and one that's to 200.
So this is within the upper 200 meters, and this is data. One data is from the upstream region, and the other one is from the more downstream region. So this is zooplankton biomass,
Right.
So
so it's everything that's taken up, everything in a vertically integrated.
Everything,
total,
yes. Yeah.
So and this was done by this is an ICES report, is that?
Yeah,
is that right?
So this data here, they do these coordinated surveys in May. Iceland, Norway, and Faroes, I think EU is part of it also, where each country cruises through their waters, and then this is assimilated into these ICES report. So this is what this data is based on. So
is there a composition information that accompanies the biomass?
No, so you don't know. And this
too bad.
It's too bad, yes. I know that they do these size fractioned data, but at least this data set that I have been working on, this hasn't been size fractioned. As far as I'm aware of, I'm hoping I'm not saying anything wrong. Maybe Hein Rune knows. But yes, but this is what they use throughout their surveys. just to estimate. So
Okay,
this data, this is being collected at the same time as when they're doing the herring surveys in May.
Yeah.
Yeah.
Yeah. Okay. So, but it may well be that this high, this high biomass is mostly Hyperboreas. Then in 2000.
You could we could anticipate that high biomasses of Hyperboreas are represented in the sample, but obviously,
Yeah.
I mean, I would guess also when you have these more Arctic conditions, I mean, the sample could also be influenced by more Arctic individuals, such as amphipods. But there's, but we don't really know. Or, I mean, we know that they're present, but you know, as I said, they're not identified what's in there.
Yeah.
But in terms of biomass, Kaldhypa is the most dominant species within the Iceland Sea.
Okay, thanks. I was just looking. So let me just get my pronunciation right. I've been calling Heinruna for 30 years, but then I heard Runa. Is there a Faroese way of saying it, or am I just? I should stick with Heinruna, right?
I'm not sure. I think he nods. Yeah.
Maybe Heinruna. You can make sure. I want to. I've been saying Heinruna for so long. Is that right?
Is it correct? Heinruna?
It's okay.
It's okay, he says.
It's okay, but what's the right way?
In Norwegian, it's heim. In English, it's more like
So Heinruna. So I guess
Okay.
you were saying it correctly. I apologize.
Well, okay. I good. So anyway, Heinruna, I thought I had another. He commented on that, and I'd just like to say, show you the biomass estimates that we have in the Northwest Atlantic. This is zooplankton biomass with a 200 micron mesh vertical net towed from the bottom to the surface, and the bottom is like 200 feet or 250 meters or so to the surface. And so the proportion of biomass, in the Northwest Atlantic generally, in Canadian waters, this is from the Atlantic Zone Monitoring Program, Catherine Johnson measurements. It's the mean fall biomass, mostly CHC5, is 12 grams per meter squared. In the Gulf of Maine, Wilkinson Basin, the mean is 16. Spring or fall, and most of that biomass, like 85 to 90% or so, is one species, Calanus finmarchicus. So the Gulf of Maine is really a refined, the Calanus life cycle just fits with the advective pattern in the Gulf of Maine. And it's really, there are a lot of similar, the role of advection. There are some similarities in what you've been studying in the Norwegian Sea, Iceland Sea, north of the Faroe Islands area with what we're seeing in the Gulf of Maine, and I might get into that. But I was wondering about, just to kind of explore more the interaction between the life cycle of Calanus hyperboreus, its vertical distribution, and the circulation and the changes in circulation in this MEIW role. And what do we know about Calanus hyperboreus, their life cycle? They produce their eggs. I assume that they're similar to in the Northwest Atlantic. They produce their eggs deep in the water, and then the nauplii swim up to the surface and wait for the spring bloom. What do we know about their vertical distribution and ontogenetic development?
Within the Iceland Sea, so Icelanders, they have studied, or they have done various studies on their zooplankton. So based on their papers, then yes, as you say, Hyperboreas, it reproduces at depth. So they think it reproduces at depth because to maximize their time to explore or to use the phytoplankton that's available within that area. And there's probably a 2-year life cycle of Calanus hyperboreas within the Iceland Sea. And that, I think, has been published, or Ingu Kristiansen, I think he's come to that conclusion in 2 papers, I think.
Yeah.
So they don't fully develop, so they stop perhaps, I can't remember, at stage 3 or so, and then they overwinter.
Right.
Yeah.
Yeah. Right. And so there's an additional complexity with Hyperboreas in that you can have interannual variations in, the relative composition of the stage in the overwintering phase, stage C3s and females.
Yes,
I guess.
We haven't really looked into that complexity, but one of the things that Cecilia Broms, who works in Norway at the Research Institute in Bergen.
Yeah, I know her.
Yes, but or she and others, they have concluded that Hyperboreas or Kallhøp is not able to reproduce in the Norwegian Sea. So, if it's not fully developed, it's very rare actually to see, stages 1 and 2, for instance. So at our section, when we have been looking at our samples, quite often you see stages 4, 5, and 6.
Right. So its life cycle doesn't work in the Norwegian Sea. Just like in the Gulf of Maine, we don't see Hyperboreas. It's coming down from Gulf of St. Lawrence, but its life cycle just can't work in the Gulf of Maine. Yeah,
that's the kind of conclusion that Norwegians, they have come to, yes. Yes, so it's similar.
Yeah, and so mostly the older, and where are these older, what's the vertical, what do we know about the vertical distribution seasonally of these older stages? You showed a really nice vertical distribution. Hein Rune showed it, of the seasonal vertical distribution of Finnmarkicus.
Yes.
What do we know about?
So the seasonal vertical migration of Hyperboreas. This is just based on this is based on studies again from the Iceland Sea, which I remember. If I remember correctly, it's residing at lower or deeper depth than Calanus or Calfinus. Mm-hmm. That's one thing, and also it's not as long in the surface as Calfin. So by I think it's June or July, Kallhøp has disappeared again from the surface.
Right, and then they go down.
And then they go
very
down.
Deep.
And then they go down very deep, I don't know, 1,000 meters perhaps or so. I think actually the overwintering depth for Calfin in the Iceland Sea, it's a maximum of 600 meters. So it's not as deep as it is in the Norwegian Sea. But I'm guessing that's probably because they find a suitable cold temperature, at a higher level compared to what they find in the Norwegian Sea.
Right.
And also, I think the maximum abundance of Kallhøp is not found in the upper 50 meters, but I think it's more found within the 50 to 100 meters.
Yeah, and they're feeding when they're up there.
So when it's in the upper layers from May to July, Calfin often we see very concentrated also within the upper 50 meters. And I think a vertical study by Ingu Kristiansen, he showed that the maximum abundance of Kallhøp is, I think it was at 50 to 100 meters. Yeah, and not 0 to 50.
Right, and just thinking of those fluorescence profiles again, and assuming that they're feeding, even though, and I don't know if there's a day-night difference in that. In the distribution, is there a deal vertical
of the
migration?
I would guess so.
So they might be up in the surface. Yeah.
Yes, but I'm not really sure if they're. I'm just thinking of this paper that Astor wrote. I'm not sure if when they're doing their diel vertical migration, whether if their upper surface layer, if this is the maximum point where they're going to, if that's 0 to 50, or if they go higher.
Right. Well, I guess I'm just wondering. It's very convincing about using CalHIP as a indicator of this, the MEIW, but just wondering if they're, trying to understand better the more details about how the CalHIP life cycle and where it is in the water column, how it interacts with where would be an MEIW. I mean, is that, so what time of year would you expect the high abundances, say prior to 2003, higher abundances of CalHIP in the MEIW? All year round, or is there like a seasonal pulse?
Well, I just kind of imagine it as a system. You have continuous flow of East Atlantic water, so the East Atlantic current is transporting this East Atlantic water. So there's a continuous flow of this, and then the more there is of East Atlantic.
And that's and that's in the surface layer, surface water, East Atlantic current.
Yeah, I guess this is in the surface layer, but also, I mean, the deeper water that's also below. So I guess the surface layer and the currents they follow, I guess, each other. So the bottom layer has a similar path as the upper layer.
Okay.
So I would imagine. I mean, I guess we're just using the modified East Atlantic water as a proxy. However, I could also imagine when the deeper water is advecting towards the Faroes or into the southwestern Norwegian Sea, and if Hyperboreas is residing within this layer, then you also get a supply of Hyperboreas, during wintertime. Yeah.
Okay.
And I guess when there's less flow or less influence of this East Atlantic water, then perhaps then it's like you don't get as much.
Okay. Okay, that's I think that's a great result that you have in this in that I guess it's highlighted in your second paper about this possibility of understanding, and I mean maybe there's and want to return to this later to check my time here, forecasting potential. And so I see I have less than 20 minutes left, but I just wanted to, so here it is. Here's what I was looking for. So, the difference between, I guess, pre-2003 and post-2003, is that right? Between the left?
Yes, that's correct.
Right. Yeah, so large Calanus and Hyperboreas, high biomass and lower biomass. So moving, just I don't know if this helps at all, but I just wanted to say a little bit about, and maybe there's some insight that you can get an understanding that the system in Icelandic Sea, Norwegian Sea system. So we have the circulation in the Northwest Atlantic coastal region. We have the Labrador Current goes around the Georges Bank, Grand Banks here, and then kind of goes subsurface and past the Gulf of Maine. We have the Gulf of St. Lawrence and the Scotian Shelf Current into the Gulf of Maine. And just noticing temperatures, the Gulf of St. Lawrence temperatures 0 to 5, or in a cold intermediate layer, and then below that, say 5 or 6 degrees year round at diapausing depths. And chlorophyll concentrations, but in the Gulf of Maine, which is at the southern margin, and which holds some of the highest concentrations, and see the biomass rivals what Hein Rune was saying in the Bering Sea in terms of the biomass of Calanus finmarchicus and the numbers. And this is why North Atlantic right whales, it's their last refuge of North Atlantic right whales, has been in the Gulf of Maine, because for some reason the life cycle of Calanus finmarchicus and the conditions of the Gulf of Maine just work. But notice the temperatures, and this is for a particularly warm year. But the temperatures can be, the overwintering temperatures can be 8 degrees or so, or even in a warm year, even higher, 10 degrees, which really accelerates, we believe, the burning of lipids during diapause and emergence and the whole timing of the Calanus life cycle in relation to the spring bloom is important. And so the finmarchicus abundance, and just this is Rubauji just published a paper this year, and the anomalies in abundance in these deep basins in the Gulf of Maine. If you just look at the right hand, the fall here, and these are anomalies, these are log scales. So there's been a considerable drop since 2010 in the abundance of Calanus finmarchicus in the Gulf of Maine. And these declines you can see especially in the eastern Gulf of Maine, in winter, summer, and autumn. It's a paper by Nick Record he published, and it's related to temperature. So the temperatures have been warming considerably at depth. These are deep temperatures, and we see less Calanus in there. And just this is in relation to North Atlantic right whales. So I don't know if you've been hearing all the troubles that North Atlantic right whales find themselves in recently, since 2010, when you have this warmer water coming in the Gulf of Maine. They're not feeding, their traditional summer feeding grounds are in the eastern Gulf of Maine, and the sightings of them and related to the abundance of Calanus finmarchicus. So the Calanus finmarchicus abundance has been down in the eastern Gulf of Maine, and right whales are no longer going there. They're going into the Gulf of St. Lawrence, and early on Canada wasn't ready for this, and right whales went right into crab fishing gear and got entangled, and there were ship strikes, and it's awful for the right whales. They've declined from 450 animals in the whole species down to about 250 to 300 animals. So it's a major concern, and there's all sorts of implications for the commercial fisheries, especially the lobster fishery in the Gulf of Maine. Anyway, this has big impact on the system, and not just for right whales, which feed directly on finmarchicus, but also on herring and other forage fish in the Gulf of Maine. And I'll go over that. And so, so now getting to herring. So anyway, I'm going to come back to like implications for future change. We're seeing it in the possibilities in the Gulf of Maine, Northwest Atlantic, and wondering about, using your studies and your synthesis here to begin to understand change in the Northeast Atlantic, or the region that you've been studying, Iceland, and the implications, what implications there are. And I think this is from your paper, your co-authored paper.
Yes.
Paper 5.
Yeah.
And so showing herring and blue whiting. And it looks like herring go down, so they have a ideal migration themselves. Is that right? And
yes. So I guess they have a similar diel vertical migration as their prey has.
Yes.
Yeah. So yes, go ahead.
No, so their feet. The implication is they're feeding on the Calanus species, day and night, or
yes. So
what do we know about that?
Well, normally you see the herring during daytime. You see them feeding at the surface, and I mean similar.
So that's these here feeding at the surface during the daytime. They're visual feeders, or are they filter feeders on Calanus?
Filter feeders.
Oh,
I believe.
Okay. I guess, yeah, they're filter feeders. I guess my time is up. So unfortunately, I'm just wondering, you know, I guess I have to wrap up.
Yeah.
I didn't realize 45 minutes. I really wanted to get into the role of lipids in the system, and we think there's a change in the lipid. Calanus has been the primary lipid contribution to the supporting the ecosystem in the Gulf of Maine, and it may be shifting. And just okay, I have 2 minutes more, and just encourage you. I would like to the role of lipids in the system is so important. You measured wax esters, and this comes back to food limitation I really like this figure here where there's the smaller females don't have much lipid at all, and then you have this great range in lipid, and how important that is to understanding the condition of herring. And I wish I could explore that more with you, but run out of time. But maybe there's more time in the future when I can get towards Havn, or we can somehow
we see each other at a conference.
See each other at a conference to continue understanding this. I think it's really interesting.
Thank you.
So I think I've done. I will complete my questioning, and that 45 minutes did go by very fast. And I'll stop sharing.
Yeah.
Thank you very much to the examiners. Now the panel will make a final assessment of the Ph.D. work and presentation and discussion here, and they will have 20 minutes to come back with a conclusion. So I think it's best that we just are sitting here or not move too much, so we are ready when the panel will come in here again. So thank you so far.
the chair for the PhD committee. We have had some great discussions about Inga's thesis, and we think that she's done an excellent job. The thesis is very interesting, and the work that has been done is extensive and scientifically sound. The thesis is a cohesive piece of scientific literature, and it has a very high standard. All three of us, I think, agree that we're very much looking forward to seeing what Inga might produce in the future, and maybe exploring more the connection between the climate change and the zooplankton community structures in the north of the Faroes, and maybe elsewhere around in this region as well. On behalf of the committee, it is my pleasure and my honor to be the first to congratulate Inga on her new title. Congratulations.
Thank you to the committee, and I'm very pleased to hear that it has been completed successfully. And here I have the diploma. So, congratulations.
Thank you.
Diploma here, and I have some roses here also for you. So, congratulations also to all your family, and all the best for your future. Hope to see many more interesting results from you. So, thank you.
Thank you very much.
And
Thank you.
after this, there is some thing on the table there for the audience.
Oh, just tack vidare. Ja. Tack vidare.